Great. So I know one person wanted to present today, but she got stuck in the middle of nowhere because the train stopped. But I think I can see one paper in the chat already. Are there any more volunteers this morning? Ooh. I was like, That's fine. Cool. Thanks for the links. Great. Welcome to Neuroccino, everyone, on this Monday morning, the 31st. We have, is that one or two papers, Lia?
Okay, no, the second link was referred to the preprint, the review, which is linked to the paper I would like to present. Yeah, I would have explained.
Okay, so that's one paper if I understood you for today. Okay, great. In that case, the rules briefly for Neuroccino, everyone is welcome to join us with or without a paper. If you bring a paper, just share the link, briefly tell us why you get excited about it, and then we discuss it to the best of our knowledge. Now, with that, I'm happy to give the floor to you, Lia. If you can start sharing your screen and then show us your paper.
Yes, so I would like to present this paper that was in my to-do list of paper to read, since as I was saying, I shared the second link that was the Neuroccino we discussed, the Neuroccino paper we discussed several weeks ago. That was the preprint review by Arnatkeviciute. It is really difficult, the name Arnatkeviciute, I don't know. And the group of Fornito, Alex Fornito, is where the genome meets the connectome. And this was one of the papers that was cited in that review. And yes, I wanted to take the time to read it and to go through this paper. So this paper was published in 2080 by Elliott and colleagues, and is titled Genome-wide Association Studies of Brain Imaging Phenotypes in the UK Biobank. So the sample that they are investigated is the UK Biobank, where you have both MRI data and genetics information. And what they try to do is link these genome informations with the they called imaging phenotypes. So it is here, the imaging derived phenotypes, EDPs, that are all the measures that you can have from MRI, starting from structural data like cortical, subcortical volume of different brain structures, cortical thickness, then also from diffusion MRI, FA of both different tracks, skeletons, and also fMRI data, resting state, task-based. So we have these three different categories of EDPs, imaging derived phenotypes, that they wanted to associate with single nucleotide polymorphisms that are some information that we can have from the genetic sequencing. And so what they are trying to do is these GVAs, that is the genome-wide association studies. So they're trying to link these two different data, imaging and genetics. And they're using the UK Biobank for doing this. They also split the data set to do like a replication studies and and what it was interesting is that they through the article also compared to the Enigma study here to see if there is actually confirmation of the previous findings about these genome-wide associations. And here there are also highlighted different websites. Now I just shared this page of the PDF. But if you click of this website, it's really interesting because you can choose the single nucleotide polymorphisms and see all the correlations with the MRA phenotypes. And so like everything is online, and there are a lot of information out there that if you are interested to, you can look into. And these plots are presenting, for example, in this figure, the readability of the different imaging phenotypes. So the variation that can be explained by the genetics information. And you can see that here in different colors, you have all the different imaging-derived phenotypes and the hereditability scale. And for example, they highlighted through the test that the volume of the different cortical regions was presenting a higher hereditability than the cortical thickness. For example, here that you see, it's like presented a lower score in the plot. Then the diffusion measures, where they also discussed that the skeleton measures were presenting a higher hereditability compared to the others. And then the fMRI measure that were actually the weakest in this analysis. So they presenting the lower hereditability with some features that were interested, but overall it was lower compared to structural and diffusion image-derived phenotypes. And these studies were in agreement with the Enigma consortium results investigated this imaging genetics relationship. So interesting in this field, a plot that is usually used is this Manhattan plot, where imaging-derived phenotypes, here, for example, the perceptibly map the T2 star in the bilateral putamen is correlated to all the genome sequencing informations. And where you see these peaks is the single the SNP that present the higher correlation with this structure. And so here you can highlight the genetic information that are more related to these imaging features. And also you can do also the inverse transformation. So on the way back to plot voxel-wise the correlations of the different genetics information compared to the spatial localization in the MRI here in the MNI template, to see also how is differently distributed the correlations compared to this mutated sequencing. For example, here, how is the stenting in the putamen, caudated pallidum, or also in the cingulate areas, to also interpret the influence of the genetic information then in the disease perspective. What else? Then they also have, for example, here they show the same thing for the in this example is a diffusion information to the crossing protein tract association to this SNP. And again, the Manhattan plot and the and then the voxel-wise correlation to interpret the different influence on so the spatial distribution in the many states of this SNP variation. And also then they explore other kind of correlations, creating these multi-phenotypes associations. So since here we are comparing a lot of variables, they created a group of imaging-derived phenotypes EDPs to correlate these groups that are, of course, correlated like in structural diffusion information together, and to try to find an highest correlation with the genetics variations. And they also explore these kind of analyses. And also they then try to correlate sometimes these features with neurodegenerative groups or schizophrenia. Also try to interpret these genetic influence in the brain. And yes, so this was this study that was like one of the main studies in this field to investigate this brain relation to MRI phenotypes, imaging-derived phenotypes with the genetics information. And they say that, of course, in this field it's really important to put together like different features that are specialized in these different fields to interpret these results. For example, one interpretation was that some genes were related to iron, and the influence of these gene variation with iron dysregulations in neurodegenerative diseases. And yes, and also they highlighted the great potentiality of these combined studies. So these metrics, it is built on imaging-derived phenotypes and singular nucleotide polymorphisms. So imaging and genetics information together in this genome-wide association view. So for me, it was really interesting because I'm trying to discover this new field, and this paper really helped me in orient myself in this quite new field in yes, in this research field. And I can try to answer to some of your questions.
Thank you, Lia. Very interesting paper. As you can hear, it's super noisy here, so I'm just going to let you deal with the questions straight away.
Okay.
Michelle.
All right. So I have a question. So it's really great. It's a really great paper. It's in open. Which journal was it published in?
Nature. Ooh,
all right. Yeah, that's what I saw. And that was 2020. No, that was 2018. So it's a paper that came out like very recently.
Yes.
Nature Neuroscience was associated genes as well with the brain. And same team, Steve Smith, and or like shoot, he published in Nature Neuroscience again.
Yeah, and I was saying that there was another paper that, as you say, there was more recent, but I wanted to read this one first because otherwise I couldn't understand the other.
So here is the question. They showed the association between genes and the brain. So there is no causation measurement in the statistical assessment, and they didn't attempt to reconstruct prototypical brains or features that would be related to specific phenotypes. That's the question.
Maybe they tried here to explain something like that in the beginning, like these partitioning redouble of functional annotations. So they maybe didn't get it because there are a lot of supplementary materials. Like here at the end, they try to put it together, both the some genetic information, no, but yes, like from the imaging perspective. So creating these multi-phenotypes associations tests. So to try to put together different features in the imaging data set, let's say, to correlate with the genetic information. So, but yes, it's not like whole brain against one genetic information. It's more like a measure, so SA or volume of different structures correlated to, so an imaging derived measure correlated to a single nucleotide SNP. It's not like let's see like all the brain to one. Yeah, I don't know if I answered.
So just to make clear, so that was low dimensional associations that were done, not high dimensional representation of the relationship between the anatomy, multimodal measurement of the brain, and genes. So that was like, you know, they're doing pair by pair. That's why they do this Manhattan chart.
Yes, there were like a lot of phenotypes. Like, yes, it's like low dimensional, if you can say, but there are like a lot of variables that they are looking to, and this is why they try them to groups. And also in the genetics information, as you can see here in the Manhattan plot, there are a lot of also genetic variables. So yes, it's as they were saying, this is the kind of matrix build up. So they are looking to one-to-one correlation. It's not like using like some features that can embed this variability. Or yes, I saw that, but I have to read it, that in the one of the papers that I cited about the UK Biobank, there was actually a principal component representation of all these SNP informations, and so the embedding of the genetics information. But here in this paper, no, it's not this.
Yeah, I have a question. So the genetic information is quite rich, and the imaging information is quite rich. How did they actually bring it together in terms of multiple comparison correction and actually associating the two statistically?
Yeah, I was thinking the same way I was answered to the Michelle information. So it wasn't highlighted in the different, in the main test of the paper, but maybe it's here in the method. I have to look about that. According to multiple, I have to look to that. Actually, of course, I look at in this kind of structure of the paper, I look more in the results, and here I look also to some of this formula that they used to look to these readability measures that I never seen before. Maybe here, genome correlations analysis. I have to look to that. It wasn't clear. It was not so clear through the test.
Okay.
I'm sure maybe they correct for multiple comparison.
There must be something. But it's a good lesson to make sure that you have the right buzzwords to search the paper quickly when we write it around. It's a good lesson for our own paper writing to put in the right buzzwords so you can easily find it.
Yeah, correction, I don't know.
Lia, there's a question for you in the chat from Eugene. Thank you for presenting the article, Lia. GWS study is great, but being an association study, it requires future studies on how to identify confounds to identify the role of a specific genetic factor. Polymorphism and environmental factors make things more complicated. It's more a comment than a question. I just realized.
Yes, no, indeed. Like, I guess that these confounding factors are taken into account in this hereditability formula, where it's supposed to modeling also the environment influencing the different interindividuality variability that we have in the different genetic phenotypes. And of course, here's an hyper simplification, but here they are trying to find the link between these genetic informations and imaging derived phenotypes. And what I found interesting is that it seems as they discussed that there is a confirmation from other studies, and also they did the replication of the results. So that gave me some confidence in this kind of correlation analysis that, of course, are like a lot of data. They can suffer about false positive, as Stephanie was discussing, and you have to be confident in things like the methods are super complicated. They are not just one like a Pearson correlation. Like it's really like you have to go there and read it. There is, I found, but they are really open. Everything is online. There are a lot of websites that they are linked to, and so I think just to take some time to go inside this new perspective that is a little bit different from what we are used from the imaging perspective, I'm saying.
Great, thank you. Are there any other questions? Yes. Oh, there you go. Go for it.
Yeah, now just on the figure 1, so where they showed the heritability of the different biomarkers. Did they talk about these? So for the functional MRI, there is these 3 green dots. So I think it's the ICA feature that are over 0.4, so 40% of heritability. Do they talk about it or?
Yes. So they saying here 4 of 6 resting state fMRI features identified by the dependent component analysis. So yes, as you were saying, that's where they are much more highly heritable. So yes, this part.
Okay, but do they give explanation of it, or is it just okay? So ICA, yeah, it means it takes a lot of the variability, so it's explaining more variance than the rest. But is there any biological explanation, something like that?
Here, what I remember that they just compare these results with the twin studies to saying that the heritability was lower compared to them, and so there was also this kind of comparison with these genetic studies. And but no, I did not remember any specific comments. Maybe there are somewhere in the supplementary material, I don't know. But yes, they were independent component features. But yeah, they more discussed the different genetics SNP that were correlating and the link with the pathologies, and for fMRI they just say that and not then explore if I can say that. Yeah, so no, they just did it.
That fully answer your question, Eri?
Right, yes.
Okay.
Yes, yes.
Just
thank you.
Quiet after. I just wanted to double check.
Sorry.
Right. Any other questions, comments for today? No? Okay, in that case, thank you, Lia, for presenting your paper, or the paper. And thank you all for coming today. I encourage you to have a paper next week, any of you. Ooh, Victor's up for the challenge. I like it. And until then, have an exciting week in science, and I see you Monday morning. Thank you. Bye.
Thank you. Bye.
Bye.
Every day I fed my brain.